Welcome to AP Biology. This video is an overview of cell communication. Cell communication takes place through signal transduction pathways. In a signal transduction pathway, a ligand, which is a chemical messenger, is going to bind to a receptor on the cell membrane of the cell. This is going to induce a series of reactions that is eventually going to lead to some type of cell response. This diagram on the top of the page shows all the different components of the signal transduction pathway. We begin with the chemical messenger, which we can call the ligand. There are three different types of ligands that we've talked about. In the nervous system, the ligand is the neurotransmitter. In the endocrine system, the ligand is the hormone. And paracrine glands release local regulators. The most important local regulator that we've talked about is the growth factor. The growth factor is a ligand that's going to tell the cell to initiate cell division. The signal transduction pathway begins as the ligand binds to the receptor. A receptor is a protein embedded in the cell membrane. The shape of the receptor is determined by the sequence of amino acids making up the protein. Each receptor is extremely specific for the ligand that can bind to it. We talked about three general classes of receptors. We talked about GPCRs, that's a G protein coupled receptor. We talked about RTKs, that's a receptor tyrosine kinase. And we talked about ligand-gated ion channels. These are receptors that specifically neurotransmitters bind to, and when the neurotransmitter binds to it, it opens the gate and allows ions to either enter or leave a cell. The pathway begins when the ligand binds to a receptor. When the ligand binds to the receptor, that message is going to get relayed throughout the cell. And relaying that message throughout the cell is called transduction. The major molecule used for transduction is going to be the protein kinase. And that's what each of these things are. Protein kinases are proteins that are specialized to become active through phosphorylation. So what's kind of going to happen here is a phosphate group is going to be passed from one protein kinase to the other. And as it goes from one kinase to the other, that kinase gets activated. So I kind of think of this as passing the baton throughout the cell, relaying the message. Sometimes we have messengers that are going to relay the message that are not protein kinases. Anything that's not a protein kinase that's used to relay the message is called a second messenger. Here you see three common second messengers: cyclic AMP, which is the most common second messenger, calcium, and IP3. These are all second messengers within the cell. Eventually, at the end of transduction, this is going to initiate some type of cell response. And that's the goal. The ligand is trying to communicate a message to the cell, and it's trying to cause the cell to do something, to trigger a response. Here you see the three most common responses, and the first one here is the most important. That's to initiate the process of transcription. We know that transcription is the first process in protein synthesis. So a very common response is to begin the production of certain proteins within the cell. Remember that proteins really run the show. Proteins do everything within the cell. So by creating or controlling the production of proteins, you're really controlling what's happening within that cell and what that cell is able to do. Another response is to change cell behavior. We'll look at that in a second, but you tell the cell to do something. For instance, you might tell a muscle cell to contract, or you might tell a yeast to generate a shmoo, which we'll be talking about in a minute. Another common response is to activate or inhibit enzymes within the cell. Enzymes speed up all the chemical reactions that take place within the cell. So by activating or inhibiting enzymes, you control what chemical reactions that cell is able to perform. Here's a picture of transduction, and here you see a series of protein kinases that are going to be activated through phosphorylation. So you see that the ligand binds the receptor. That causes an initial relay molecule to be phosphorylated, and this phosphate group is passed from kinase to kinase. We call this series of phosphorylations a phosphorylation cascade. It can sometimes involve second messengers as well, and eventually this is going to lead to some type of cell response. Here we see some pictures of responses. In this picture here on the left, the ligand right here is going to be the growth factor. Growth factors are proteins. They're polar. So any ligand that's polar is going to bind to a receptor on the cell membrane. Then you see transduction take place with a phosphorylation cascade. And the last protein that gets phosphorylated is going to enter the nucleus and it's going to work as a transcription factor. Transcription factors activate genes. They turn genes on and begin the process of transcription. Transcription is going to lead to the production of a protein. Here you see another common response. It's the same response but just triggered by a different ligand. Here we see a steroid hormone like testosterone. Steroid hormones are non-polar, so they go right through the cell membrane. Since they go right through the cell membrane, they bind to a receptor within the cell. Once they bind to a receptor within the cell, that complex enters the nucleus and again works as a transcription factor. It's going to activate a gene and initiate the production of a protein. Steroid hormones like testosterone and estrogen and progesterone regulate the process of protein synthesis. They activate genes and they control what proteins are being made. Here you see an example of a change in cell behavior. This behavior is going to allow yeast cells to mate with each other. In order for yeast to mate, they have to generate a structure called a shmoo, which is this projection sticking off of it. Once they generate a shmoo, the two yeast cells can fuse together. So you have to induce a change in the shape of the yeast. So our ligand is a mating factor, it's the name of it. It binds to a G protein coupled receptor, causes a G protein to get activated through phosphorylation, and that begins our phosphorylation cascade. Now the last protein to be phosphorylated, its job is to produce actin microfilaments. Actin microfilaments make up what we call the cortex. Actin microfilaments that are right underneath the cell membrane, they give the cell its shape. So by increasing the amount of actin filaments, essentially what's happening right here is that you're changing the shape of the yeast cell, creating this shmoo, allowing it to then be able to mate and fuse with another yeast. So here you see another common response, and that common response is a change in cell behavior. That concludes this video.